We have to finish by 4:15 because there is again another meeting at 4:15. Yes, you can proceed now.
Okay.
Govindwar sir, proceed.
Yes, I'm not getting this. Okay. Yes. Can you enlarge the screen here? Because I'm getting very small screen here. Can you make it full screen? Oh no.
Medha, is it possible? To enlarge the screen?
Sir, I think using mobile phone is not possible to enlarge the screen, sir.
Okay, so whatever you can show, earlier you are showing that you can show like that.
Sure. Just a minute.
And your voice is breaking, Medha. Echo is there. Yes. Sir, now is it visible to you?
Yeah, it is visible to me, but I do not know how to go. Anyway, just wait. I can enlarge this.
Phytoremediation technologies for clearing.
Okay, I got it.
Yes, current developments in bioengineering and second slide. Okay.
Oh yes, just wait. Yeah. Okay, this is. I think you are changing the slides.
Yes, second slide.
Okay.
Current development in bioengineering and biotechnology.
Yeah. This is what I say. Those who are working on degradation, and they want to know that exactly what is degradation and what study is being done till to date. Because particularly this book, this is the series publication by Ashok Pandey, and we have published this current developments in bioengineering and biotechnology. That is a book series, and one of that book is Advances in Eco-Friendly and Sustainable Technologies for the Treatment of Textile Wastewater. And this is mostly edited by our group. Okay, that is myself, my student Mayur Kurade, and Boyang Hyun John and Ashok Pandey. And if you are interested to know that exactly what is degradation and what study is being done 2022, it is covered here. Okay, next slide. Next, please. Okay, so you're so hurrying. Just wait. I can put. Can I get earlier slide? Hello, 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 Dr. Deshmukh.
Sir, when I am presenting, it's not possible for me to get connected audio.
Okay, you can proceed.
So I can't keep my mic on, otherwise there will be some disturbance. So sir, whenever you want me to change the slide, please let me know. I'll keep on changing the slide.
Yeah, sure. Please give me slides.
Yes.
Okay. I will start from here. Now, this is a present scenario wherever the rules and regulation are not followed strictly. Particularly, we can find all over India, this is happening. Most of the wastewater is throwing in the rivers, and rivers are getting polluted. And you might be seeing that particularly few newspapers, they are giving now the polluted water in Sangli and Ichalkaranji. Okay, this is the scenario. But only question is that where the rules and regulation are not being followed. Next, please. Next slide, please. Okay, so this is what is the, can I get earlier slide? Okay, okay. This is water pollution, mostly the problem, and why we should solve the water pollution problem is because of the toxic textile dyes are very toxic. They are carcinogenic, mutagenic, allergic, cytotoxic, and genotoxic. And present scenario, we can remove that with the physicochemical methods. In that method, particularly adsorption on the because coagulation, adsorption, all these things, you can produce a non-degrading sludge here, and that non-degrading sludge goes for landfilling, and landfilling also after some time it will be leaching because of high concentration of dye polluted at one place only. Then you can find the discharge too. That is possibly people make that dye is available for the environment. Next, please. And so it is a serious concern of water and soil contamination. Next, please. Next, okay. Now, what are the once I do not know. I would earlier say. Okay, just wait. Potential bioremediators, those who are working on dye degradation, they should take care of what are the biological sources they can do the dye degradation study, that bacteria, fungi, and the plants. And mostly the tiny microbes, chemical processes, it has unlimited power working together to clean up the messes of the man. That's why most of the things as microbes are very important for the cleaning of the textile wastewater. Next, please. Next. Now, what is that? How you can achieve this? We know that microorganisms do degrade the textile dyes. That can be achieved by microorganism, by adsorption. This is particularly adsorption and biotransformation. These are the techniques. But you can use the microbes, that you can adopt microbe, you can genetic manipulate microbe, and you can use specific microbial biotics for the remediation of the dye. Next, please. Okay, these are the treatments, biological treatments are available, which works in presence of oxygen, and this is aerobic treatment. In this treatment, now presently we are using this. Color is usually not destroyed, might be absorbed on the biomass, and degradation of organic pollutants. This takes place using the biological treatment. Okay, and that treatment is in presence of oxygen. Frequently, this method is used in CETPs. Next, please. Another way. This is the aerobic process. What happens? Organic pollutants consumes oxygen and nutrients, microbes, and you get the carbon dioxide and H2O, and oxidated products of the molecule. Next. Oxidation, particularly oxidation processes are taking place. Another is an anaerobic treatment, which takes place in presence of O2. In this case, the production of methane, CO2, and water requires less energy, can break down the compounds with the aerobic process, which cannot break down like that. And toxicity of degradation products is there. And mostly, a best example is azo reductase. Most of the azo bonds are breaking in anaerobic treatment. Next. Okay, so these are the different microbial remediation, textile microbes. Particularly, we have studied these microbes for the degradation of the textile dyes. Okay, these are the number of microorganisms. You can find this whatever, if you go find the Scopus, you can make all the publications from my laboratory, which shows the microbial remediation of the textile dyes. Next, please. Okay, so yeah, this is the slide that those who are working on dye degradation, what we need to do. In short, I made a flow chart of dye degradation study, how one can go for the study. This is for mostly plants. I have mentioned screening of various aquatic plants and for phytoremediation. You can say the bacteria, those who are working on the microorganism, they could go for the bacteria. They can isolate different type of the bacteria. Then they can evaluate the decolorization process in presence of bacteria, what happens. Then most important part is the enzymes are very important here, like lactase, lignin peroxidase, tyrosinase, azoreductase, NADS, DCIP reductase, veratyl alcohol oxidase. These are the enzymes responsible for the metabolism of the dyes. Then to check that the compound, the dye is getting metabolized or not, then and what active groups are present, that you can go for FTIR, GCMS, HPLC, HPTLC, like that. And on the basis of the CMS results and the enzyme assay, you can predict what is the pathway for dye degradation. After degrading the dye, you can check the toxicity, like phytotoxicity, MTT assay on aquatic animals and on molecular techniques. And ultimately, I have mentioned here constructed wetland. Naturally, if you go work with the plant as well as work with the microorganisms, it is naturally you can go with the wetlands. Because presently, if you are going for the phytoremediation and the microbial remediation, wetlands are far better than your CETPs. Next, please. Okay, this is for those who are working in that, you can find in conical flask, there is a removal of the color. And in another spectrum, you can find out that what is the degradation. This is the constant. Can you see the arrow what I am putting here? Can anybody answer me? There is a decrease in the peak at 600 nm, and that is because of degradation of the dyes and degradation of dyes here. So this happens, and only just by without looking towards the flask, you can say that yes, there is a degradation of dyes. Next, please. Now, this is a potential of the microorganism. This I have added some because Dr. Megha said me that most of the students are there, they are working for the dye degradation study. That's why I say here, one should be aware of that different type of the bacteria has a different pattern of decolorization. I have put here time versus decolorization, and there are 4 types of the bacteria available, and their rate of decolorization is different. That you should understand. That's why we have to study with the number of bacteria. Otherwise, if you know that bacteria is doing this business, you need not go. It is not generalized statement that bacteria does that, but the potential of doing metabolizing the dyes is different with respect to the bacteria. Next, please. Next. I'm not getting. Next, please. Okay. Yeah. Yeah, this is the very important slide. Can you make it clear, because I couldn't see that complete slide. Can you go back and check the slide, because I'm not able to see the slide here. Earlier slide. I have to go for earlier slide. Megha, can you change the slide? Yes, okay. Yeah, I should say about to Lakshmi now here. Lakshmi, if you see here the graph. Next, please. Okay. Now, in this slide, you can find here, there is a Congo malachite green, cotton blue, methyl violet, and crystal violet. But malachite green, if you see that up to 10 minutes, 10 hours, you will find this continuous decrease of malachite green, right? But you can't find any decrease in crystal violet. In crystal violet, sudden decrease is there, there is no further decrease. Same case with cotton blue, there is a decrease, but sometime after 5 hours, there is no decrease. This indicates that cotton blue, crystal violet, they are absorbing on the yeast cell. However, malachite green is degrading here. That you can find that it is getting absorbed or degrade. For absorption, there is a sudden decrease in the, you can find sudden decrease in the level. As in case you can say here, in malachite green, there is a sudden decrease and then slowly degradation is there. And that means that this sudden decrease is because of the absorption, and gradual degradation is for because of the degradation of this. Just by this, you can understand that the dye is getting degraded or absorbed. Next, please. Okay, this slide is giving you that whatever the source you are using, the potential source. Because earlier people used to say that degradation is because bacteria are not getting the food material. That's why bacteria is going for the degradation and getting energy from that chemicals. That was the earlier thought. Okay. Now, yes, it is correct. If you do not have food material, one has to go for the looking for different type of the foods. And yes, bacterial cell can do that. That's why we checked with the glucose and sucrose over here and urea and all these things. Yeah, if you add this, there is a change in the degradation pattern of malachite green. Okay, so here the most of the concept what we are using is bacteria is not getting food and it is getting degraded. Okay, that is one thing. And within this concept, we have seen earlier that what will happen in presence of glucose, what will happen in presence of sucrose, what will happen in presence of molasses, what will happen in presence of urea, it is nitrogen source. And we found that, yes, there is a variation in the degradation pattern. Next, please. And it is same with medium composition. There is a change in degradation pattern. Next. Okay, now here you can find that different concentration of dye is being used. What is the time required? As you increase the concentration of dye, you get more time. Then what is the bacterial mass you are used? As you increase the bacterial mass, time is decreased. Then different medium gives you different potential of the degradation. Temperature also has an effect, as well as pH is also have an effect. So these are the basic studies one has to do earlier. But nowadays, with this study, I don't think that you get the publication. Next, please. Next slide. Okay. Now here we are coming to the view that earlier we said that the degradation is because of the less food material, but it is not. There are two ways. One is bacterial cell is looking for the food material, as well bacterial cell is looking for their safety. And because of the chemicals present in the environment, bacterial cell dies. And to protect by their own, they produce some enzymes, and that enzyme is taking care of the degradation. Okay, there are two different concepts we are following. Okay, earlier concept was dyes are working as a food material. So in presence of food material, degradation was not there. In absence of food material, there was a degradation. However, another concept what we are seeing is that if security concepts, presence of dye gives you the security. The presence of dye gives unsecureness to the bacteria, and bacteria is producing the enzymes, and that enzymes is taking care of the degradation. Now, in this case, you can say it's a simple biochemical experiment. It is with the laccase, that in presence of dye, you'll get the competitive inhibition. And competitive inhibition is because of both are the substrates. Laccase substrate is also present, and whatever dye you have added is also the substrate. That means that the dye which is present there is substrate for the laccase, and that substrate is getting degraded. Okay, and that's why we have to go with the enzymes. Security concern. Next, please. And slide is not clear. Medha, slide is not clear. Now, okay. Now here we are seeing here lignin peroxidase, laccase, azoreductase, malachite green reductase, NADPase, DCIP reductase. Okay, and we are seeing the percent declaration also. So what will happen and what time is required for the when it is getting decreased at different temperature also we have work on that. If you see that green color words, malachite green reductase, we have found one of the enzyme there. Lignin peroxidase, what will happen, what will be the percent degradation, what is time required in different temperatures. And if you see here that I have marked with the green, if you see that, that is an induction. Enzyme is getting induced. Lignin peroxidase is getting induced at 50 degrees centigrade. Laccase is induced at 30 degrees centigrade. Azoreductase induced at 5 degrees centigrade. So you can find here, effect, particularly this experiment was performed with the what will be the effect of temperature on the dye degradation. Okay, but you can understand that different type of the enzymes are getting induced at different temperatures. Next, please. Next slide. Next slide. Okay. Now here, don't change now. Now here you can see here what will happen. As I say that, no. You should go back. Medha. Medha, can you listen to me? I want earlier slide. Earlier slide. Earlier. Now, in this case, mostly the dye degradation, textile wastewater we are using, number of salts are also present. And in this case, we have studied the number of salts. What will be the effect on dye degradation because of the salts? Now, in this slide, you can find that always it is not possible to use glucose and fructose and sucrose. I think there is some problem that I'm not getting exactly the slides. Okay, now I've got it. Now this is hello. Hello, is there any problem?
Yes, there is a network connectivity problem, sir. Just a minute, sir. I will share the PPT with the one person who is having the laptop so that you can present it.
Yeah, I do not know what is the exactly problem. It is better that if I myself upload this. If I say share screen, it is not possible for me. I think I should be the one of the
Yes,
also. I should be one of the
you
hosts.
are co-host, sir.
Huh?
Yes, you are. You are one of the co-hosts, sir. You can present your screen from there. Because of network issue, it's getting disconnected. Shall I share the screen?
Yeah. You can share the screen. There is no problem.
Yes, sir. So if you can let me know the slide number you want, I can just find out so that accordingly I can run.
No, just you change slide one by one only. You are changing the slides 2-3 times like that. One by one. So that will be better for me.
Ma'am, it was 24 slide number.
Wow.
Ma'am, I think he wanted 22nd.
But I'm not, I do not know exactly. Where is your screen? Yeah, I got it now here. Yeah, earlier slides. Yeah, this is the slide I want. Okay, now in this slide you can find that as we say that different enzymes are responsible for the metabolism of the dyes. Here we took two different microorganisms. So one is the yeast here, galactomyces geotrachum, and bacillus species. And you find here that the green color, azoreductase, in case of bacillus species, you can find azoreductase and tyrosinase. This is increase in tremendous enhancement of this enzyme activity over here. In case of galactomyces geotrachum, there is no increase in the activity. So in combination, in consortium, you'll find all lignin peroxidase, azoreductase, and tyrosinase are increased. And that's why you can get 100% degradation within 2 hours. Okay, so in short, we are showing here that the enzymes are responsible and the combination of the enzymes works for degradation. Next, please. Okay, in this slide, you can find here that it is not possible to use that the glucose and the fructose or sucrose all the time, not necessary to use. So we have to go with the agriculture waste, and this experiment was performed to go for the agriculture waste to reduce the cost of the processes. Next, please. Next, please. Okay, this is we are working with the environmental concern, and mostly that is containing metal ions. So we have to check with the different metal ions what will be the effect of metal ions. Now here in this slide you can see here, in one side we are getting different dyes, and we have worked with the only one Brevibacillus latorospirus here and different enzymes, and you will find that different dye increases the different type of the enzymes. You can see the green, all green things there, and what is the different dyes. That is the mechanism for the metabolism in the different bacteria is different. Next, please. Next one. Next slide, Meda. Hello, next slide, please. Yeah, now you hear that these are the, how you can check with, these are the different microorganisms, pictures are there, and you can see the decrease in the peaks just by using the spectrophotometric study at maximum wavelength. You can look for degradation pattern. And you can go with the TLC, simple TLC can be used to see that, yes, there is a metabolism of the compound. Next, please. Effect of pH is there in the metabolism, and not only the reduction in the color. You can see that at control there is only one dye at pH 6. You got the metabolite, but at pH 9 you got more metabolites. Just simple TLC can work for the showing that yes, there is a degradation. And a question from the Lakshmi, you can go with the TLC and check that how many spots you are getting. Next, please. Another way to look for that, you can go with the HPLC patch. HPLC, here there are different patterns of the HPLC. You can see that the control peak at what retention time it is getting, and increase in number of peaks that gives you the indication of there is a production of the metabolites. Okay, next please. Again, you can go with the FTIR. FTIR mostly gives the particularly that what type of the groups are available, and active groups are available. So if you take a FTIR of the control sample, and after treatment, if you take a FTIR, you will get the difference. And that means that yes, there is a degradation of the dye. Next, please. Yeah, it is okay. Same, please. FTRI, you can go with the FTRI. Next. GCMS is a very important study here. And GCMS gives you what different types of the molecules are generated in the sample. And on that basis, you can go with the flow chart for the probable pathway of the metabolic pathway of the dye, how the dye is getting degradation. With respect, if you consider that enzymes, the particularly lackases or reductases are increased, they will support that your pathway is correct or not. Next, please. Now, this is we try to go with the propose a pathway for brown 3R REL. Now you can see here that we have shown is a cleavage with the peroxidase enzyme, and you got the product. And this is whatever the structures you will find, that is because of these structures, molecular weight particles we got in our effluent. And that's why we propose that, yes, probably this might be the pathway for that. And that is why it's a proposed pathway. So what type of the metabolism is taking place there? Next, please. Yeah, this is also okay. Next, please. This is only metabolism, and this is on the basis of the GCMS and your GCMS release and enzymatic activity. Next, please. Same case with lignin peroxidase. We got the product, and we got these 2 peaks, 2 or 3 peaks here in GCMS, and we can say that here breakdown of a particular bond here. Okay, so it is lignin peroxidase. Lignin peroxidase works for that. Next, please. Okay, so this is the important for me. Now if I say that what will happen, suppose the dye is inserted in the bacteria, and what will be the products? And if you see here, the HPLC pattern of all these 3 are different. Okay, so and that is because in Cochlear rosea, Mg reductase is induced, and you will get the different pattern of the metabolites. Brevibacillus lattosporus, lackage is induced in higher quantity. You get the different pattern of the metabolites, particularly I mentioned in GCMS. Then Saccharomyces cerevisiae, Mg reductase is induced. You'll get the different pattern there. That's why if you get the induction of a particular enzymes, that will lead to different fate of the metabolism of the dye. Okay, so if you take different type of the bacteria, product are different, even though same compound is there. Okay, if you take malachite green and degrade malachite green with the Cochlear rosea, Brevibacillus lattosporus, Saccharomyces cerevisiae, products are different. And it is essential that we have to look for the non-toxic product. Okay, so that's why we have to take care that what type of the bacteria are to be used to get the non-toxic product. Next, please. Next. Next slide. Hello. Yeah. Next slide. Something is wrong. Okay, this is, as I say that the product should be non-toxic. We have to look for the what type of the bacteria or the fungus you have to take. And here the usual that phytotoxicity you can study here. And you can find here the central one is a red color, that is a dye, is a red dye, you can say. And we are using here sorghum bicolor. And you can see here the A, B, and C something that control what will happen, and after treatment what will happen. You will find that control what without dye, with dye, and dye metabolites. You'll find that dye metabolites, in presence of dye metabolites, plants grows in a normal way. That means toxicity is not there. Next, please. Next slide. Then you can go with the microbial activity, what growth, what is the effect of that compound in the microbes, and you can find that what are the circles are getting formed is normal antibiotic study. You are studying that effect of antibiotics on microbial growth, like that. What is severity of the antibiotics to grow in the microorganism? The same study you can do here. Next, please. You can go with BOD. Mostly if you're talking about the environment, we have to take care of the BOD, COD, TDS, TSS. These are parameters are very important for us. So If you're talking about the environment concern, you have to go with the BOD and COD. Next, please. Yeah, same case, COD should decrease here. So with the time goes, COD is getting decreased. I think you must be aware of that biological oxygen demand and chemical oxygen demand. Next, please. Then what will happen? The chromosomal what will happen with the chromosome comet assay? That this assay gives you that the is there any effect of dye on chromosomes? Okay, chromosomes of allium cepa we can use here, and you can get damage is taking place or not. If you would like to see that decrease in the potential of that dye to degrade the chromosomes, you can go with the comet assay. Next, please. These are the another result of the comet assay. You can find this type of the structures, and you can see here what will happen with different dyes. That is another way to look for the toxicity. Next, please. Okay, now here we were talking about the only bacteria, because bacteria are involved in the metabolism of this particular compounds, and we are CETP for anaerobic bacteria and aerobic bacteria, and that degrades. Now the concern was that there's only bacteria is useful for the metabolism of these dyes. But one thing I should be sure here, whatever the technology which is present in India for CETP, they are the best technologies possible, but rules and regulations are not best because they are not treating the chemicals properly. So that is the major drawback, that we couldn't control any environmental pollution. Okay, that I should be very clear. And supporting, mostly what happens, if you use the bacteria, there is no decrease in TDS, total dissolved solids, and that is a major problem. So if you use phytoremediation, then you can decrease the TDS. Okay, and for that, this is the studies what they did. We are in our laboratory. We started that. I think mostly, if you look for in the scopus, degradation of textile dyes by phytoremediation of textile dyes, you will find number of papers from our laboratory. More number of papers from our laboratory, where this we have started here. And we thought that plants are take caring of degradation of dye. And so we have proved that. Certainly we have to prove that, yes, plants are doing that. First, we should we get the plant cell culture, Blumia. We have shown that there is a metabolism of dyes. Then callus culture also we have shown there is a decoloration of the dyes. Whole plant system also Brassica we have took and shown the decoloration of dyes. Then hairy roots also we have took, that is mostly transgenics. There also we have shown the decoloration of dyes, and we have shown that yes, plants are involved in the metabolism of textile dyes. Okay, next please. And on the basis of that, here there are number of plants what we have studied which shows the degradation of dyes. Okay, there mostly you'll find that these research papers are from our laboratory, but there are few groups in Turkey as well as Pakistan. They are also working on the phytoremediation of textile dyes. Next, please. Okay, now here, why we are working in the phytoremediation? Now here you can find here combination of these plants and bacteria works better. Now here in this you can say number of dyes I have shown here. Time is also there. Consortium means one is the plant and one other is a plant and another is a bacteria. So sorry, these are the two different plants. Two different plants as I shown in bacteria, there are different bacteria works in their own way. So if you combine two bacteria together, that is a consortium, will give better performance. It is the same thing here. If in consortium, you'll find 100% degradation, but these two plants are showing that less amount of degradation. It's a magic only because some enzymes are induced very well in one plant, some enzymes are induced very well in another. So that is the magic what we obtain in bacteria, same case in the plant consortium. Next, please. Okay, now this is, now you can understand here that what happens. In first table, you can see that yellow marks. Laccage is induced in Gaillardia pulchata and Amaranthus alus. You can find a different enzyme level, it is increased. Two different enzymes in the same, they are increased in two different. Laccage is one, and another aster amylase, you will find that veratriol alcohol and lignin peroxide is induced in a lot. Okay, the reason that is why that the rate of degradation was less. But in consortium, because both are there, so you will get the induction of lignin peroxidates, veratriol alcohol oxidates, laccage. And because all enzymes are increased here, you will get the better performance, 100% degradation within less time. Next, please. This is the proposed pathway. Is the same thing what we propose the pathway on the basis of the enzyme present. You'll find that the two different plants gives two different metabolites, and consortium will give different metabolites because of the composition of the enzymes present in the plant. Next, please. Okay, so why we studied the plant? The answer is here: plant and bacterial consortium. You know, mostly that CETPs are available and to degrade the dyes because of the bacteria. But in this case, you'll find that plants also degrading as well. If you find in at 56 hours, you'll get the degradation of the complete degradation of the dye because of the consortium. If you go with the consortium, you will get degradation is faster because here bacterial enzymes as well as plant enzymes are working together and you are getting more degradation. For example, I can say here particularly about the laccage. Laccage is not that active. Plant laccage is not that active for the degradation. It requires the mediators, redox mediators, but bacterial laccage doesn't require redox mediators. So in combination, in plant lackages are more in concentration. However, plant lackages are less in bacterial concentration. But if we go in the combination, redox mediators are generated because of the bacteria and this metabolism. So both lackages work properly, and you will get the degradation is more faster. Next, please. It is the same story we can get with the Xenia angustifolia and Exobacterium bacteria and the plant composition. You'll find lignin peroxidase is increased in Xenia, and bacterial azoreductase is increased here. So whatever you will get, the combination of these two. And laccage is in both the conditions. Now here in Xenia angustifolia, lignin peroxidase is increased. This is the plant. And in bacteria, azoreductase increase. So because of the combination of these, lignin peroxidase as well as azoreductase are working here. So you get the metabolism faster. Okay, and ultimately the composition of enzymes is important for the metabolism of the dyes. Next. It is the same what will happen in the consortium, different type of the metabolites you will find. In bacteria, you will get some type of the metabolites. In plants, you will get some type of the metabolites. In consortium, different type of the metabolites. Depending on that, what of the metabolence and what the toxicity, you can choose that you have to use the bacteria as well as the plant. Next, please. These are the reactors what we have developed. And in this reactor, you can find that both bacteria can grow in the soil as well as plants are also there. And you can find in whatever the red circle, what you are decrease in COD, and you can say the metabolites with different dyes we have mixed together, and there is no metabolite found. No metabolite found over here. That means plant and microbes consortium work. But this experiment suggests that combination of soil microorganism. One thing is very important. You might be aware of that the microorganism can be cultured or non-cultured microorganism also there. But if you take a soil bioreactor, in this reactor, non-cultured microorganisms are also present in the soil. So both uncultured or cultured microorganism can work here, and the combination works very well for the metabolism of that. Next, please. So here, because we are talking about the environment, parameters are taking BOD, COD, ADMI is gives the indication of the color. These are all parameters, and you will find here TDS. Especially, I am interested in TDS. There is a decrease in the TDS because of the plants. Okay, it is very important to have the phytoremediator, phytoreactor to decrease the TDS. Normal CTP plants, you will not find there is a decrease in the TDS. Next, please. This is the another study that we checked the different type of the bacteria they grow in the soil reactor, and what will the enzyme system is working and how you can get the metabolites. Okay, next, please. This is a simple experiment what we did, that what will mostly, you know, if you're talking at the removal of the dyes from the water resources, not necessarily, because soil is also getting polluted, and these plants can grow in the soil, and you can get removal of that. Here you can find here that we have grown this Pulchetta galladia in presence of Pseudomonas bituta. The fourth number, if you can see here, the plants are green. In third number, it is without bacteria. So microbial source also supports for the growth of the bacteria. Okay, next, please. This is the another reactor we have developed, that in first reactor, we have the plants only, and in second reactors, we have the plants which are growing in the soil. So in second reactor, you will find here that the plants as well as microorganism is taking part for the metabolism of the crop. Next, please. Okay, all the time we are talking about the HRTS, and HRTS, to develop the HRTS is very difficult. That's why it is one of the attempts we made here, that what will happen if you make a vertical system. What you might be having the concept of vertical gardens. Similar techniques can be make here vertical reactor and circulate that textile effluent vertically, and like this you can make in one room you can make a vertical reactors like that, and you can clarify the sample, and it is possible you can make it. It is just like your vertical gardens. Next, please. This is the HRTS and the phytoreactors we have developed at Kolhapur, Kolhapur MIDC, Kagal MIDC, and this you can say that the plants are also there, soil microorganisms also we have used, and we have used for the remediation purpose. This is the active plant which is present in the Kolhapur. Next, please. Here also you can find that particularly flowering plants we have used here. Soil degradation, that is, you can say that dyes present in the soil that can be degraded by the plants. And in second chart, which is present down, you can see that in presence of the plant, dye level is less. If in absence of the plant, dye level is increased, because this is only accumulation. And in presence of plants, different type of the microflora will be developed. Plants also are taking care of the, that's why you can get decrease. In upper, you can see here, that whatever the content of there, that is dye content is also decreasing. Water is getting purified. Okay. So you can use the flowering plant to make a beautiful atmosphere there. Okay. Next, please. These are the all parameters what we have seen, that different plants what they will do. All parameters like phosphorus, nitrogen, potassium, what will happen to the soil. Okay, so certainly it is minimizing the pollution, inorganic ion pollution, as well as this phosphorus and potassium, that is also. Okay, next please. These are the, you can say, all other parameters we use, what will happen in the consortia. And in consortia also, we got the results that there is a decrease in mostly TDS we are bothering about, and TSS, this phytoremediation we are bothering about, TDS, ADMI value is also decreasing. So phytoreactor is certainly working better. That is taking care, microbe as well as the plants are taking care for the degradation. Next, please. Now again, question comes that whenever there is no land, so you can go with the floating beds, and this you can say in first picture you can say that the roots, how we have developed the roots, and these roots are taking care of the degradation of the dyes. And you can find here in the table there is a decrease in ADMI value, COD value, BOD value, and TDS also. So everything is decreased. So now what we are proposing here, the last chamber of the CETP, they can grow phytoreactors or phytobeds there, so you can get the decrease in the TDS. For your information, in present situation, at last chamber, there is no decrease of TDS. If you go with the phytoremediation, you can get decrease in the TDS there. Next, please. Okay, this is what we propose, that the phytoreactors, that is a microbial source as well as the plant, from the industry to the river, if you make this type of the channels or nalas, growing with the plants as well as the having some good soil bacteria, whatever the effluent is coming through the CETP plants or the industry and goes to the river, that will be clarified during their path. Thank you very much.
Thank you, Govindwar sir,
Okay.
for in detail description of the azo dye degradation, and many young generation, many scientists, young scientists, they are working on this azo dye and they want a guidance.
One thing is, I can say that although it is not advisable to say to you, but I sent that book e-copy to the Megha, so she can circulate personally to the members, those who are working here. I have sent e-copy to you, right? E-book.
Yes, I will do the business. Sure.
Yeah. So only
Very
that.
nice.
Only question is that don't circulate too much. Only your friends you circulate. Those who are working.
Correct.
Because that is the
Correct.
copyright problem is there. This copy what we got as a, because we are the authors or the editors.
So what I suggest, Medha, it's
Yes, sir.
4:18 because I have to shift to another lecture now.
Yes, sir.
So what I will suggest you,
Yes, sir.
next Sunday, you
Okay.
arrange only discussion, no presentation of the Dr.
Okay.
Sanjay Govind.
Okay, sir. Fine. Sure.
Only discussion will be there, question and answer.
Sure. Yes,
So
sir.
those who
Sure.
are interested will join.
Okay.
Okay, and
Okay.
now come express vote of thanks because I want to finish it and shift to another. lecture.
Sure. So it was really a very nice session, a very knowledgeable session. First of all, I would like to say sorry for all the technical glitches that we had in between the sessions because of the network issues. But we are very thankful to you, Dr. Sanjay sir, for such a knowledgeable session that you shared your experience on biodegradation along with the result analysis as well as the descriptive explanation about all the things. I feel all our friends will have got very nice opportunity to go through the entire research what is done, and I will ask all friends to approach with respect to their queries for the next session which we will be conducting on next Sunday. So thank you again, Dr. Gurulima sir, Dr. Gurulima sir, as well as all the participants for attending today's session.
Again, there is a one question in chat box. That is the give email ID of Govindwar sir. Already he has given in the chat box.
Yeah, already I gave.
And it's very simple. spgovindwar@gmail.com. Very simple.
And we will share the mail ID even in the group, so you need not have to worry. We will share it in the group as well.
Correct. And next time, next Sunday, there will be only discussion with Dr. Govindwar sir. Thank you.
Okay, well, just check that I do not know exactly what is next Sunday is the first, okay? 2nd of April.
No, with your permission, we'll circulate.
That's why, because I'm not sure that where I am.
Okay. Maybe next Sunday, maybe after 15 days.
No, just
But
check with
we'll
the
have
next
only
Sunday.
one full discussion.
Yeah, sure.
No presentation, only question and answer.
Right. No problem.
Okay, thank you.
Meanwhile, all participants can study my slides, and if they have any question, they can ask on that too.
Yes. Now, today we'll circulate this YouTube.
Yeah, that is a presentation as well as, yeah, no problem.
Okay, thank you.
Okay. Thank you very much.
Thank you.